Vegetative propagation. In plant body, we have been studied the flower. Flower is the reproduction part of plant, but the remaining part, leaves, roots, stems, they are vegetative part of plants. So an organism which is similar to parents if formed by using stems, leaves, or roots of plant, that kind of reproduction, we call it vegetative propagation. So vegetative propagation is a common type of asexual reproduction in many plants and ferns. The plants have vegetative propagation in many ways, by stolons, like in strawberry, rhizomes, tubers, corms, and bulbs. All these parts are vegetative part of plant. They are not related to sexual reproduction. The sexual reproduction part of plant is flower, but this part they act in vegetative propagation. There are three general types of vegetative propagation, as natural vegetative propagation, artificial vegetative propagation, and tissue culture for plant tissues. So the first one, natural vegetative propagation, may happen in many ways, like propagation by stolons. In one of the vegetative propagation types in strawberry is an example for that kind of reproduction or propagation. A horizontal stem which can be a meter in length extend above the surface of the ground. A plant, strawberry plant on the surface of ground in that form. And the stem of the plant is herbaceous plant, so it not stand, but is extend on the surface of soil. After a time, it makes roots at the connection points of nodes. These stolons form a new vertical plants on the node position on the stolons. Vertically also they produce stems and make a new plant. Again, the stem extend on the surface of soil. The node point touch the soil, and below it makes root. Above form leaves and make a new plant. So a new plant, which similar to mother plant, is formed, but by using stem, not sexually part flower. So a name of the stem which extend on the surface of soil is, we call it stolon, that we call it propagation by stolons, and strawberry is an example for this kind of propagation. Propagation by rhizomes. Rhizome also a vegetative propagation way, which most of the weeds and ferns reproduce by this way. It happens by formation of terrestrial stem. Again, a plant, but this plant has like soil surface, but the plant has an underground stem. So in rhizomes, the stem is under the soil, but closer to the surface. In node position, again, it vertically form leaves and form roots. Extend node position above, make leaves at the below. It makes roots, and a new plant similar to mother plant is formed by using stem, but because stem under the soil, that kind of terrestrial stem, terrestrial means totally covered with soil, we call them as rhizomes. Roots grow from horizontal stem, and green parts grow from the buds above the ground. Terrestrial stem extend by the growth of their apical buds, which contain apical meristematic tissue, cover new areas in great speed, like in garden grass or iris. Garden grass is a good example for this. The green part of grass which we see, they are leaves. But we pull the grass, simply we can see the white colored terrestrial stems and tiny roots under the soil but closer to surface. Propagation by tubers. Tubers can be defined as food storing terrestrial stem. They are again terrestrial, means they are covered with soil under the ground, but as a different from the rhizomes, they store food. Tubers contain number of lawns called eyes. The good example for tubers is potato. So when we hold a potato, on the surface of potato we see some lawns, they call it eyes, and this eyes they contain, each eye contain bud or many buds, call it axillary buds. These buds, they contain apical meristematic tissue which new stem, extend, stores food, and expand to make a new tuber. Again, on surface there are lawns called eyes, each of them bud, stem, extend, make a new tuber, stores food and expand. Usually one plant has a group of tubers, maybe 1 and 2 and 3 or more, which are capable to form new branches from its bud, as in potato. Each of them on surface have the lawns colored eyes, which are axillary buds, contain apical meristematic tissue for extension, new branch, and then new tuber, like this, to make a group of tubers. Terrestrial stem like potato. Propagation by bulb and corms. Bulb and corms. This kind of vegetative propagation happen in many herbaceous plants, by herbaceous plants which have no secondary growth. A bulb is a single globular beak, but is a simple definition for bulb. It has a disc stem at the base, and some leaves grow from its upper surface, roots grow from the lower surface. The best known examples for it, the onion, garlic. In onion, the big part of onion that is being eaten there, leaves. At the base of these leaves, there's a disc-shaped stem, and the lower surface, it makes tiny roots, where they are leaves. Buds are emerged from the axis of leaves, and from the connection point of leaves to the stem. Their growing points contain axillary meristematic tissue. New buds can form from it in that form to make a new onion, a big globular bud that we call it bulb. In garlic also, only one slice of garlic when we plant under the soil, again made up of number of leaves, a disc stem with tiny roots. From each axillary bud, a new bulb may grow. So after a time, we may see 10 or 12 or more number of bulbs for one just garlic. So each piece of garlic, we call it that was one bulb, but a complete onion also one bulb. So it may separate or remain connected to the mother bulb. Onion, garlic, narcissus, lily are examples for this type of propagation. In corms, also method of vegetative propagation, similar to bulbs in morphology and in appearance, they look like one another, but difference in big part of corm is the stem tissue. So big part of bulbs in this tissue, leaves. But in corms, most of the organism is stem. A big part of stem, like what? Like earth apple or turmeric or ginger. And this big stem covered with tiny leaves, which can be separated from it. Tiny leaves cover the big stem, and book axillary position from this nodes position, and new corm mains grow, again covered by tiny leaves. Tiny leaves can be separated or removed from the surface, but most of part of this plant which being eaten or stored the food is stem tissue, while in bulbs most of the plant which is being in stored food is leaves. The leaves are thinner and smaller than the leaves of the bulbs. Corms also reproduce by forming buds from the axil leaves, then stems separate to form new corms and gladiolus, earth apple, and turmeric. So there are some differences between bulbs and corms. In bulb has big leaves which stores food, but under the base a disc-shaped flat stem. Most of plant which stores food being eaten is leaf, while in corms most of its stem which covered with tiny leaves. And also we said bulb example as onion, garlic, but for corms gladiolus, earth apple, or turmeric. Artificial vegetative propagation. Many plants lose their ability to form active seeds, like in banana, grape, and some kind of orange. And also some plants take a long time to reproduce by seed, like dates, which takes many time to grow and make seeds, also to give fruits. And also there is difficulty to guarantee the limit the species, because in sexual reproduction, gamete formation and cross-pollination always change the quality of products. And the genus of tree, therefore farmers resort to propagate the plants vegetatively, which we said is asexual reproduction. So by asexual reproduction, the new organisms formed from the same mother plants are same, with same quality. So if a farmer wants to preserve the quality of products, need to produce new plants by propagation, but not sexually reproduction. There are some plants cannot reproduce vegetatively. They need stimulation to propagate by using some plant hormones. Indole acetic acid, indole butyric acid, and naphthalene acetic acid are three hormones which is needed to activate the plants to reproduce vegetatively. Offsets, an example for artificial vegetative propagation. Offsets are big buds frequently formed at the stem base in the region of its connected with the soil. So in an date palm tree, the connection point of stem which connected to the soil, new plants like plantlets grow. These plantlets, each of them we call them as offset. So these plantlets, the adventitious roots which extend in soil are formed from them. When their growth completes, they are separated from the mother artificially, handmade. Then they are translocated and grow in another position to grow into a complete independent plant. Among plants which are propagated by this way are date palm, which is common in Iraq, and banana. Layering. Some plants can form adventitious roots if they touch the earth and cover it with soil. Soil surface, so some kind of plants, especially some herbaceous plants, which their stem are flexible, they can be bended at the connection point with soil. It makes roots and leaves, and then it can be separated from the mother plant, transported in another position, planted, and grow into new independent plant. Man has utilized this characteristic in propagating some plants in that a twig, a small branch is bended, with the stick connected with mother plant and part of the covered with some quantity of soil, and this kind of vegetative propagation, formation new organisms similar to mother, but using stem, bending, touch the soil, produce root, we call it layering, artificial vegetative propagation. Another type of layering is aerial layering. if the bending of the branch is difficult because of the hardness, for example, it can be layering by enclosing with a wet soil. A woody plant has stems and many branches. So, but because the stems has secondary growth, so solid cannot be bended. So, by enclosing with a soil containing pot, a branch of plant covered with amount of soil in a pot on a plastic bag. Water is given to the plant for about 6 weeks. After 6 weeks, the node position, it starts to produce roots and new leaves. As roots and leaves formed, it can be cut from the mother animal, translocated in another plant in soil, and it grows into new plant. Among plants which can be propagated by laying is grape or lemon or orange. Grape for layering, but lemon and orange are for aerial layering. Why I call it aerial layering? Because the twig or branch is not bended to be touched the soil, but in air it covered with amount of soil in a plastic bed with given water to form it adventitious roots and then be separated from the mother plant to be planted with another position by hand. So we call it example for artificial vegetative propagation. Propagation by grafts or grafting. The process of grafting is attachment part of plant to part of another plant. This method is used to reproduce plant with desired properties. It is not said in the experiment if a part of plant is taken which has buds, orange, it has bud in a special way on another plant similar to which has similar characteristic. And guess what? There are two plants, one the lemon tree and the orange tree. So a small twig is cut from the orange and transported to be set on the lemon tree, fixed on it, tied firmly, and wait for it to grow on the lemon tree, but this part produce orange while the remaining part as lemon tree. The sign as stock, special scientific names for these two kinds of plant. The stock, we use this term for the plant which is connected to the soil, like lemons in this example, but the scion is the plant which part of it is transported to be attached another plant like orange. So in this position, orange is the scion, but the lemon is the stock. So for grafting, two types. Budding grafting. A bud is taken from a plant with desired properties, like an orange tree, and placed in a split of T-shape within the stalk. Again, stalk, the plant which is connected to the soil. The external tissue, which we call it cortex, in T-shape is cut. So it's not totally, but it's separated from the plant, not to open. Then a twig, a branch which has many buds from another plant is transported, set in it. The parts are covered again, tied firmly and covered with wax substance, make it to grow with the stalk. So by transformation, a bud from the scion to the T-shaped split which is taken on the body of stalk, new plant grow. This kind we call it budding grafting. Cleft grafting is carried out by cutting the stem of the stalk 3 horizontal near the surface of soil. The stalk 3 horizontal parallel to the soil. The stem totally is cut. Then vertical splits are made in it. 2 or more vertical splits can be done. Afterward, a branch containing number of buds taken from the scion, from the second plant. It is trimmed with the end of it is sharpened like this in such a way that fits the split of the stalk. After it fixed in the stalk plant in that position, tied firmly, covered by waxy substance to prevent infection. So covered with wax can be asked and as a cause question. One, then this place is tied out, covered with wax. The reason was covering with wax is to prevent affliction or infection by parasites. It is important to know that grafting always not succeed. Only if there are alike properties which are genetically look like one another or similar, only can be attached on one another. Means they must be from the same class of plants. So orange cannot be grafted on peach, but can be grafted on lemon. Also peach can be grafted on plum. So an important point during settlement of scion on the stalk, we have to take care of cambium tissue. Cambium tissue is the tissue between xylem and phloem. So when cambium tissue fit on one another in stalk and scion, means xylem and phloem also stuck on one another. So because if these not fit one another, xylem and phloem of scion cannot receive food or water from the stalk, and maybe that time the grafting is not succeed. The importance of vegetative propagation in plants and why farmers do the vegetative propagation, or how much is important the vegetative propagation for plants or in agriculture. To propagate the plants which do not produce seed, means they cannot reproduce sexually, so they being reproduced by asexually propagation. To propagate the hybrid plants without change, because hybrid plants which formed by cross-pollination with good quality, if farmers wants to preserve the quality of their product, they produce new plants by propagation. To propagate the plants which seeds are germinated at low rates, or propagation the plant with speed up giving fruit, it takes much time, like in date palm tree. So we make them to reproduce artificially by offsets. To adapt the plants with new conditions, like what, for example, roots of the pear trees do not grow in sandy soil, but in peach they can grow in sandy soil. So we use the stalk of peach tree, but the scion from the pear tree, so pear tree can grow in new conditions without any problem. The sixth, then preventing the affliction by some parasite, we attack the roots of kind of some plants, but not attack some other kinds. As an example, the roots of European grape has been afflicted, means infected by type of parasite, which this parasite not infect the American grape. So if the stalk of American grape part, the plant which is connected to soil, are grafted by what? By the European grape. So the fruits are from European grape, but stuck from American grape, and European grape can grow in this kind of soil, which contain parasite without any problem. Plant tissue culture. The plantation of plant tissue is regarded one of the application of vital techniques which have in reproduction of plants, and this represents the artificial vegetative propagation. Simply it means the development of plant tissue or plant cells outside the plant body in environment like laboratory, in proper feeding media. Proper feeding media means a media allocation or a liquid or mixture which contain all necessary materials for a tissue or cell to survive. This results in formation or development of plant buds which transform during the proper time form a mature plant. Means what? Some amount of cells are taken from a plant and help them to grow in laboratory into new mature complete plant. The advantage of this tissue plantation in plant is the method uses apparent time, so to produce the plant with the desired properties, which is aim of the vegetative propagation too. The salt resistance and the change in the temperature, and also overcoming plantation problems, which decrease the succession of growing new plants in environment, but by this method they make plant they grow in laboratory as a certain time and then transported to the environment. The tissue plantation is becoming common in world and also in Iraq. Some studies and research centers achieved a success in this field, and this can be the main reproduction of date palms, which are commonly grow in Iraq. So we study as example for tissue culture in date plants. So how this tissue culture or by tissue culture method new plants are formed in laboratory? In 4 steps, one of the offset we remember we said in plants which like banana or date palm, new plant let's grow at the base or point which mother stem connected to the soil, and we call them as offset. So the offset is being separated from the mother plant, then the apical meristematic tissue, apical growing part extracted, separated from the plant. Why? Because it contains active cells which have active growth. This requires sterilized media to prevent contamination of extracted tissues. Why? Extracted tissue means other tissue, which is permanent tissue, have no ability of division. The apical growing part partitioned into small pieces, transported into laboratory, small partition into smaller pieces in that form, because it contains active cells. The tissue planted in media like test tubes, which contain necessary materials for plant tissue to grow, and some about each of these apical cells or active growth meristematic tissue cells transported in from each these tubes, and after a time these cells, like germination of seed, they grow into new young plants. As they produce leaves, start to do photosynthesis, then after they transplanted into normal environment, a new plant similar to mother plant is formed, but artificially in laboratory in a medium which contain all necessary materials. We call it as tissue